Welcome to Department of Botany, Serampur Girls College. Today we discussed about pteridophyte and our topic is life cycle of Pteris. Myself, Dr. Madhusri Haldar. Let's start the life cycle of Pteris. First of all, we have to know the sporophytic plant body of Pteris. The sporophytic plant body of Pteris is differentiated into root, rhizomata, stem and leaves. The primary root is ephemeral and is replaced by a large number of adventitious roots developed all over the surface of rhizome. That is, the primary roots degraded after some time and adventitious roots are developed from the surface of the rhizome or stems. The rhizome or the stem may be creeping in case of Pteris grandiflora or erect in case of Pteris vittata, which may or may not be branched. Rhizome is differentiated into nodes and internodes, and its entire surface is covered with scales. Next thing is that the growing point of rhizome is covered with ramenta. Ramenta is nothing but dry brown colored scales. The leaves that born on the upper surface of the rhizome, when young, the leaves are spirally coiled and shows circinate vernation that is typical of true fern. Leaves are called pinnae or pinnule. They are pinnately compound with a long rachis or midrib. That is the sporophytic plant body of Pteris, and in the upper side that is the circinate vernation. The pinnae are small near the base and towards the apex, while they are larger in the middle portion. Throughout the world, almost 250 species of Pteris are distributed, but in case of India, some species are found like Pteris vittata and Pteris criteca, etc. Next important thing is the rhizome or stem anatomy. Anatomically, the rhizome shows three different layers. The first layer is the outer epidermis. Next, the second layer is a few layered thick sclerenchymatous hypodermis, and the innermost layer is the broad parenchymatous cortex with diversified stellar organization. That is the transverse section of rhizome of Pteris that shows the outer epidermis that is cutinized or cuticle is present on the outer surface of epidermis. Next to the epidermis there is a hypodermis, and then the parenchymatous cortex, and in the cortex region there are many stellar organization. The stele is very much diversified because different kind of stele is found in Pteris. Some may be solenostelic. In case of Pteris grandiflora and Pteris vittata, it is solenostelic. In case of Pteris criteca, it shows dictyostelic configuration. But in case of Pteris biaudata, different region of rhizome shows different types of stellar organization. That is, when a cross section of the lower portion of the rhizome shows a mixed protostele, the middle portion shows siphonostele, while the apical portion of the rhizome shows the polycyclic dictyostele. So we can see that in a single species, the organization of stele may vary. In general, the stele is made up of a number of meristeles forming two rings. The innermost ring consists of 2 to 3 large meristeles, while the outer ring comprises of a number of small meristeles. Each meristele has a band or plate-like mesoxylem surrounded by its endodermis. Next important thing is the anatomy of leaf. In leaf, there are two different regions. One is the rachis or petiole portion, and another one is the leaflet. The petiole portion shows distinct anatomical view. The first layer is cutinized epidermis, then sclerenchymatous hypodermis, next to that parenchymatous cortex, and then uh, there is leaf trace of various shapes like C, V or U shape embedded in the endodermis, and that is the transverse section of rachis of Pteris. When the cross section of the leaflet shows an upper or and lower epidermis layer, and the lower epidermis consists of stomata. Next layer is the mesophyll tissue, not well distinguished into spongy and palisade parenchyma, and innermost layer is vascular bundle. And that is the anatomical view of transverse section of the pinule or the leaflet of Pteris. Next come to the reproduction. When we cut a mature leaflets or pinule of the Pteris, we find something about the reproductive structure of Pteris. So, Pteris is a homosporous fern, that is, all the spores are structurally and functionally alike. Next thing is, all the fertile leaves bear sporangium aggregated to form sorus. Sori are represent sori are present in a continuous line on the edge or margin of pinnae called sinosori. That is, when the sori are present on the both margin forming a continuous line, that is sinosori, and the sori actually borne continuously on a vascular commissure connected with the venens. Next, that is the picture of the sinosori. Here with the arrow shows the continuous line of brown colored mature spori. Sinosori are protected by the reflexed margin of pinnae called false indusium. Sori are mixed type because both mature and immature or young sporangium are present in the same sorus, intermingled with many sterile hairs in between the sporangium. That's why the sorus of Pteris is known as mixed type of sorus, and that is the transverse section of the pinule of mature pinule of the Pteris, because it shows the sporangia and the hair-like structure and also the false indusium. The development of the sporangium in case of Pteris is of leptosporangia type. When a sporangia develops superficially from a single cell, it is considered to be as leptosporangia type. Let's look on the structure of a mature sporangium. This picture shows the mature sporangia of Pteris. One is top view and another one is side view. A mature sporangium has a long stalk that terminates in a capsule. So the sporangia of Pteris is stalked, and the capsule layer, which bear actually the spores, are jacketed. In jacket there are three different types of cell: a thick walled vertical annular cells complete that actually protect the sporangial structure, a thin-walled radially arranged stomium, and then the third type of cell is large parenchymatous cell with undulated walls. So the capsule contains many spores, and spores are triangular in shape with trilate aperture. The sporangium dehisced transversely along the stomium due to the shrinkage of annular cells, and the spores dispersed in the air to moderate distance. So main function of the stomium is to dehisc and dispersal of the spore, while the main function of annulus is to protect the sporangia and spores. Let's have a look on the life cycle of Pteris, because in case of Pteridophyta there are distinct alteration of generation, and that is diploid and haploid phase is very much distinct. So that is the mature sporophytic plant body of Pteris, on which pinnately compound leaves are there, and leaves which bear the sporangium are known as sporophyll, that is 2n in, or their chromosome number is diploid, and that bear mature and that bear sinosaurai on the both edge of the leaf. In each sorus there are many sporangium that are also 2n in number, and from each sporangium spore mother cells are produced, that is also 2n or diploid chromosome number. Each spore mother cell produce spore by meiotic cell division or reductional division. Hence the chromosome number of each spore is haploid, and from the each spore gametophyte is developed, and that is also n number of chromosome, and from each gamete and within each gametophyte archegonium or female reproductive structures are there. They have egg within it, and within antheridium or male reproductive organ there are sperms. After fertilization of egg nuclei and sperm nuclei, zygote is formed, which is diploid, or chromosome number is 2n, and from this 2n zygote young sporophytes will develop on gametophytic plant body. So that is the complete life cycle of Pteris. Here we can show the distinct two phase. One is the sporophytic phase or 2n phase, and another one is gametophytic phase or haploid phase. That is all about the life cycle of Pteris. I think that the video is very much helpful for the student. That's all. Thank you.